All right, let's jump right in. Today, we are taking a really close look at a virus that's a perfect example of a Dr. Jekyll and Mr. Hyde story in virology, the John Cunningham virus.
And that right there, that's the central question we're going to tackle. How does this silent, incredibly common virus, this companion, suddenly transform into a devastating pathogen that attacks the central nervous system?
You know, for most of the population, this virus is just a harmless part of our personal virome. We pick it up in childhood and it just lives with us. But in a way, it's like a ticking clock. Because under the right, or I should say the wrong conditions of immunodeficiency, it reactivates, and the results are catastrophic neurological diseases. So let's break down exactly how that happens.
Okay, so here's our roadmap. We'll start by exploring its two-faced nature. Then we'll get into its core biology. After that, we'll trace its journey from a dormant state to a lethal one, see how we track it in the lab, and finally, we'll talk about the immunological battle we're up against when we try to manage it.
So first, let's really cement this idea of duality. It's a concept that pretty much dictates our entire clinical approach to this virus.
I mean, just look at these numbers. The prevalence is absolutely staggering. We're talking up to 90% of adults carrying this virus, and for them, it's a complete non-issue. The whole thing changes. That pathogenic switch gets flipped only when the host's T-cell-mediated immune surveillance just collapses. And that completely reframes the problem, doesn't it? The virus isn't really the primary aggressor here. It's the host's failing immune system that opens the door.
So what are we actually dealing with on a molecular level? Let's profile the fundamental biology of the virus itself. Formally, its name is human Polyomavirus 2. It's a member of the Polyomaviridae family, which puts it in the same club as the well-known BK polyomavirus. Its genome is this compact, circular, double-stranded DNA molecule. Now, the early and late coding regions, they're pretty conserved. But the really crucial part, the part you have to pay attention to, is the non-coding control region, or the NCCR. This region is hypervariable. Think of it like a molecular switch. It can undergo these rearrangements that allow the virus to change, to adapt from its quiet, latent state in the kidneys to a highly replicative, brain-invading form.
Structurally, the virion itself is a classic icosahedral particle. It's small, it's tough, and, this is really important, it's non-enveloped. This lack of a lipid envelope is key to its stability out in the environment.
And this is a perfect way to see what we're talking about. On the left, you've got that naked capsid of a virus, like the John Cunningham virus. On the right, an enveloped one, like herpes virus. Without that fragile, fatty outer layer, the John Cunningham virus is just way more resistant to breaking down, which has real implications for how it gets transmitted.
So how does this incredibly stable virus go from being a silent, lifelong infection to a lethal neurological disease? This next section breaks down that exact pathogenic journey.
It all starts pretty innocently, with an asymptomatic infection, usually in childhood. This leads to a lifelong latency, primarily hanging out in the kidneys. The trigger for disaster is always a severe drop in cellular immunodeficiency. That's the signal that allows the virus to reactivate, spill into the bloodstream, and then, critically, cross the blood-brain barrier and just wreak havoc in the central nervous system. Now, once it's in the CNS, it doesn't just cause one disease. And this is a fantastic example of intra-host evolution. Depending on which specific genetic mutations occur, the virus actually changes its target. The classic disease, PML, involves that NCCR rearrangement we talked about, which lets it target oligodendrocytes. But if you get mutations in the VP1 gene instead, its target can shift to cerebellar granule cells, causing GCN. Other mutations can lead to encephalopathy or even meningitis. It's incredible, really.
Okay, so this brings us right to our doorstep in the laboratory. The modern diagnostic workflow here requires a really crucial distinction. Understanding the purpose of each test is absolutely critical for us as clinical microbiologists. Because using the wrong one at the wrong time, well, it can have very serious consequences.
So here's the bottom line. We use two completely different tools to answer two completely different questions. For an asymptomatic patient who's about to start a high-risk therapy like natalizumab, we use serum serology to figure out their risk. A positive antibody test tells us they have the latent virus and are therefore at risk. But for a patient who is already symptomatic, serology is useless. For them, we need a quantitative PCR on their cerebral spinal fluid to prove active viral replication inside the CNS.
And here's how that all fits together in practice, specifically in the Dutch context. CSF qPCR is our standard of care for an active diagnosis, period. Serology is the standard for risk management before starting drugs like natalizumab.
And notice, viral culture is strictly for research. It's just way too slow, it needs very specific cell lines, it's too fussy for any clinical use. And histopathology? That remains our definitive gold standard when the PCR is negative but the clinical suspicion is still sky high.
Okay, this brings us to our final section, management. And it really truly is an immunological battle, because after decades of research, we still have no direct weapons against the virus itself. And a huge part of that is the immense challenge of getting effective drugs across the blood-brain barrier.
This quote really says it all. The core principle of management is this. We spent decades searching for a direct-acting antiviral, and we've come up empty. Survival depends entirely on the host's ability to restore their own T-cell response to fight the virus in the brain, which, makes the whole question of antiviral resistance mechanisms kind of a moot point. We simply don't have an effective therapy for the virus to develop resistance against in the first place.
And that strategy has to be tailored to the root cause of the immunosuppression. For HIV, it's about starting or optimizing combination antiretroviral therapy. In the Dutch context, for natalism-associated PML, it's a very protocol-driven process. Stop the drug and begin urgent plasma exchange. This lines up with international guidelines from places like the CDC, which also put immune restoration above everything else. For kids, the principles are the same. It's all about reversing the immunosuppression. But of course, all the interventions are tailored for pediatric doses and physiology.
And this is how we have to think about prevention. We need to be very clear. We can't prevent the primary infection, it is just too common. We prevent the disease by preventing the reactivation. And that is all about careful immunological stewardship. It means using risk stratification before therapy. It means monitoring lymphocyte counts during therapy. And just making smart clinical decisions all around.
So where do we go from here? Well, the path forward is pretty clear. The Holy Grail is a safe, effective, direct-acting antiviral that can actually get into the brain where it needs to work. A second, but still crucial goal would be a prophylactic vaccine to prevent the primary infection in high-risk people before they even get to the point of needing immunosuppressive therapy. And that brings us to the end of our look at the John Cunningham virus. It's a story about a virus, its host, and the incredibly delicate balance of our immune system. The big question for all of us remains, in an age where we have more and more potent immunotherapies, how are we going to manage the silent pathogens that are already waiting inside our patients? I would love to hear your thoughts on that in the comments, and please subscribe for more deep dives just like this one.